皆様、リンク J のライブウェビナーにご参加いただきありがとうございます。本日はリンク J UC サンディエゴジョイントウェビナーシリーズ第8回 with 慶応大学再生医療と計算神経科学における最新の展望セッション4ステムセルズ&キャンサーをお送りいたします。私は本日司会を務めますリンク J の関と申します。よろしくお願いします。本日は画面にお示ししているプログラムに沿って進めてまいります。ご質問はウェビナーの Q&A 機能からお願いいたします。時間の都合上、すべての質問へお答えできかねる場合がございますので、あらかじめご了承ください。また、本イベントではウェビナーの同時通訳機能、英語字幕をご利用いただけます。なお、ウェビナー中の録音、録画、画面キャプチャーなどはお控えいただけますようよろしくお願いいたします。それでは、リンク J 常務理事曽山と UC サンディエゴ国際アウトリーチディレクター和賀様よりご挨拶いたします。よろしくお願いいたします。
はい、皆さんおはようございます。本日はですね、当ウェビナーにご参加いただきまして誠にありがとうございます。今お話ありましたようにですね、UC サンディエゴ校とリンク J はですね、これまで UC サンディエゴ校、そして、日本のですね、例えば慶応大学、大阪大学、京都大学と、トップ大学のトップの研究者の方々にですね、ご登壇をいただいてですね、ライフサイエンス領域における様々なテーマを取り上げてですね、ウェビナーを共催してまいりました。そして今回はね、シリーズの8回目、となります。UC サンディエゴ校のターニシュタ・レア先生、そして慶應義塾大学の佐谷秀幸先生にご登壇をいただいてですね、幹細胞とがんについてご講演、プレゼンテーションをしていただいて、その後ディスカッションをしていただきます。また、両校のですね、アンドリュー・マクロー先生、そして岡野秀幸先生にはモデレーターを務めていただいておりますので、よろしくお願いいたします。そして今回はですね、日本の視聴者の皆さんの中にですね、高校生の方が多数参加されております。慶應義塾大学のご講義によってですね、JST のプログラム、こちらを通じてですね、高校生の皆様にもお声掛けをして、ご参加いただいているということで、ぜひね、興味を持っていただいて、Q&A もございますので、遠慮せずに、おじさん、おばさんに遠慮せずにですね、どんどん手を挙げて、質問、書き込んでいただければなと思いますので、よろしくお願いします。そしてこのシリーズを含めてですね、リンク J、UC サンディエゴ校とコラボレーションしているんですが、とてもですね、それを助けていただいて、いつも大変、ご助力をいただいている UC サンディエゴ校オフィスオブリサーチアフェアーズのですね、インターナショナルアウトリーチディレクター、和賀さんにですね、今日もご参加をいただいて、おります。和賀さんからですね、まずはイントロダクションとですね、モデレーターの先生方のご紹介をよろしくお願いします。和賀さんよろしくお願いします。
Hi, konnichiwa. Good morning, Japan, and good afternoon, California. I'm Miwako Waga, Director of International Outreach at UC San Diego. It's a pleasure to continue to host the joint webinar series, inviting excellent speakers from UC San Diego and Keio University. I'm happy to report that at UC San Diego, the fall quarter started last month with in-person class sections accounting for about 80%. The positive cases on campus remain very low, although just about 20% of campus employees are working on site. Yet it is now possible to host small in-person meetings on campus. Actually, I recently hosted a meeting with a pharmaceutical company executive from Japan with a faculty member on campus. I do hope that international visits will resume in earnest in the near future. I encourage you to start making plans to visit California, perhaps in early spring 2022. Now let's ask Dr. Okano, Professor and Chair, Department of Physiology at the Keio University School of Medicine, to give brief remarks about the collaboration between the two institutions, and Professor Makalo, Director of the Institute of Engineering in Medicine at UC San Diego, to make some comments and introduce the first speaker, Dr. Tanishita Reya. Okano Sensei, over to you.
Okay, thank you very much, Waga Sensei. It's my great pleasure that I can make the announcement of this webinar between the UC San Diego and Keio. Actually, so 2015, Keio University School of Medicine or Graduate School of Medicine made a sign up with MOU to collaborate with the UC San Diego Health Science. And so it was renewed last year. And we extended this collaboration, including the School of the Engineering in UC San Diego and the Faculty of Science and Technology in Keio. Now you can say that so medical science and engineering, and we can Keio and UC San Diego work together. And so for this purpose, this regenerative medicine and neuroscience is one of the best topic for us. And so in terms of this, today we focus on cancer stem cells. And so we invited Professor Hideyuki Saya from Keio, and so Professor Tanishita Reya from UC San Diego. I hope that both speakers will give an excellent presentation for all of you, indicating the cutting-edge science of the cancer science and also cancer stem cells. I hope you enjoy this webinar. Thank you very much.
Hi, konnichiwa. Arigato gozaimasu, Kana sensei. Good morning in Japan and good afternoon on the West Coast. I am Andrew McCulloch, Director of the UC San Diego Institute of Engineering and Medicine. And it's my pleasure to add my welcome to this webinar on New Horizons in Regenerative Medicine, jointly organized by Keio University and UC San Diego and generously hosted by LinkJ. My special thanks to Miwaka Waga-san from the Office of Research Affairs at UC San Diego and Akihiko Soyama-san from LinkJ for making this webinar series possible. We have two really outstanding speakers for this fourth session in our series, which is devoted to stem cells and cancer. And it's my special pleasure to introduce the speaker from UC San Diego, Professor Tanishita Reya. Dr. Reya is Professor of Pharmacology and Medicine at UCSD, where her lab works on the signaling pathways that regulate cell fate, especially between stem cell renewal and commitment, and how these mechanisms can go awry and lead to cancer. She's widely recognized as a world leader in this field and has received several very prestigious awards, including the Ruth Left Siegel Award for Pancreatic Cancer Research, the Frederick Ailey Award for New Discoveries in Immunology, and the National Cancer Institute Outstanding Investigator Award. I'm really delighted that she can be here to speak to us today on the topic of stem cell signals and the origin, progression, and therapy resistance in cancer. Tanisha?
Hi, thanks so much. So should I go ahead and share my screen?
Yes.
Yes, please. Yeah.
Okay, great. Okay. Well, it's a special pleasure to be part of this, and I really appreciate the invitation because I've been actually collaborating with faculty at Keio, including Dr. Akano and Dr. Suda, for over 10 years. And so I think I've really benefited in a lot of real ways, as you will see as my talk sort of unfolds. So I will talk generally about how stem cell signals are hijacked to drive cancer progression and therapy resistance. And I'll just start generally with this idea that one of the things that really drove us to ask these questions is how pre-malignant lesions become malignancies, because most of us will have a pre-malignant lesion. It's sort of like having a mole. These are fairly differentiated and they're very slow growing. And in and of themselves, they are not a threat to anybody. But with new mutations, this goes on to becoming a real malignancy that we all know of as cancer, often incredibly therapy resistant and often difficult to control. So we have really focused on what changes between the pre-malignant state and the malignancy. And in context of this, we have noted that the pre-malignant state is often differentiated and the malignant state is often immature or undifferentiated.
And so we have focused on this concept of the differentiation arrest that occurs as a potential place where malignancies become as aggressive and therapy resistant as they do. You can think of this as development in reverse, because normally as you're growing, you go from having immature cells to mature cells. And in this case, it's as if the mutations have unraveled all the programs that were put in place so carefully to turn this into a very aggressively growing set of immature cells. In thinking about this as development in reverse, over the last many years, we have focused on how stem cell programs may contribute to this, because in normal circumstances, stem cell programs are high in development, extinguished as development progresses, but in cancer early on in pre-malignancies, these programs are low, and they become more aggressive and become more dominant as the cancer becomes more aggressive. And so we've really focused on how these stem cell programs may contribute to the aggressiveness of cancers. And in this context, I'll say that where we're trying to go long-term is that if we understood the programs that were important to drive this into malignancies, it may be useful for early detection, because these pathways are aberrantly activated when they shouldn't be. And they also may be important for interception, because if you understood what drives this into a malignancy, you may be able to stop it sooner, because as all of you know, that the cancer field has done a lot of great work. We know a lot of pathways. We even have a lot of great drugs. But more often than not, we always detect cancer too late. So really our long-term view is somehow to move all of this back earlier, so that all of these tools that we're developing can be used at a point where it can be successful. And just as an example of how developmental signals can be useful, we published a paper on hedgehog signaling in myeloid leukemia in 2009. And based on that and other work, Pfizer did a trial on hedgehog inhibitors in myeloid leukemia. And this inhibitor, called Durismo, was approved for use in de novo AML a couple of years ago. So this is a really great example of where a very basic project that we were doing was taken up and moved forward into the clinic. And it's actually something, a pill that a cancer patient actually takes. So it's been a source of great inspiration to really continue this work. So the story I'm going to tell you today really starts with our observation that NUM, which is a pro-differentiation molecule, is high in early malignancy, pre-malignant state, but is downregulated as this becomes aggressive and immature. And what we were trying to do at the time we were at Duke University is try to understand why NUM was going down and how this was contributing to making these cancers immature. And so I was very fortunate to stumble onto work that was done by Professor Craig Montel and Professor Hideyuki Okano at that time at Hopkins. And what they had done was find this this program called Musashi, or this gene called Musashi, and they had mutated it, and they found that upon mutation, these single bristles that you find in Drosophila become double bristles instead of a bristle and a neuron. And they had sort of the foresight to name this after the iconic Japanese samurai named Musashi, who fought with two swords. And this, I'm sure, is, you know more about this than I do, but I often think about this as one of the best scientific decisions they ever made, because it really helps me out in my work and also in terms of my talks. So we focused on this partly because of Dr. Montel's work and Dr. Okano's work following that, showing that this may be an important regulator of stem cells. And we asked whether this might be important in cancer. So what we did was we looked at a slow-growing cancer called chronic myelogenous leukemia and compared it to an aggressive cancer called blast crisis, found that Musashi, just like it's up in stem cells, down in differentiated cells, it's reverse, where it's down in slow-growing cancers, and it's very upregulated. In fast-growing cancers, if you block it, you gain survival. And if you look at patient samples, these are 120 patient samples, 90 from the Hutch, you can see that in patients, chronic disease is very low for Musashi, and that it accelerates with accelerated phase and in every blast crisis patients is incredibly high. Along with this, we also found that not only does it rise as the cancer becomes aggressive, but that it might also be a prognostic as patients who are in chronic phase, if they have upregulated MSASI, they are actually at a high risk of relapse or death. So this is something that has been followed through by other labs in other cancers. And so in general, we think this may be a prognostic as well as a potential therapeutic target. This is just a quick summary of that part, showing that when the first hit occurs for myeloid leukemia, this is BCR-ABL, we think it triggers proliferation survival, takes it to a chronic indolent phase where MSASI is low, NUM is high. But with the second hit, what happens is MSASI is upregulated, and that downregulates NUM and other programs that allows these differentiation arrests to occur. But the good news from all the work was that this could be a potential therapeutic avenue because it wasn't stuck in a different undifferentiated state, and that you could increase differentiation either by shutting down MSASI or by elevating NUM, and that could block leukemic growth. Around that time, we were actually in the middle of moving to California, to UCSD, and we had noticed that besides heme malignancies, MSASI was actually very high in gliomas, in high-grade breast cancer, and colon cancer, and so on, but it wasn't clear if it had sort of any functional role there. So we were really interested in knowing if this was sort of a paradigm controlling really aggressive recalcitrant cancer. So we worked at UCSD with a surgeon, Andy Lowy, on pancreatic cancer as one example of solid cancer that's very, very untreatable. So it used to be a fourth leading cause. It is now the third leading cause after breast cancer. The treatments are radiation chemotherapy for large tumors and surgery for small tumors, where only 20% of the patients are actually candidates. But at the end of all of this, the disease does return, so that the survival rate is one of the lowest you will see in most of oncology, and it's at about 9%, 9 to 10% in the last few years. So there's a lot of work to be done. So there's clearly a lot of clinical need. But one of the things that drew me to this question, although I was coming in from leukemia, which is very different, is that actually, if you look at how pancreas cancer develops, early on, there are these pre-malignant lesions with early mutations, and they are dysplastic, proliferative, but quite differentiated. And with later mutations like P53 loss, these cells become really aggressive, become really immature and undifferentiated. And this is really where the lethality is. So although we were studying a very different system, in principle, this cancer was doing something very similar. So we worked with Andy Lowy to look at patient sample resected from his own patients. In red, you can see MSASI 1 or MSASI 2 heterogeneously distributed in the cancers. At end stage, it's often acutely expressed across the cancers. And with the help of many people, what we did was we crossed the MSASI knockout, both 1 and 2, with the 1 being from Dr. Okano, into a model for pancreas cancer called KPC. This is just showing you an MRI of a normal pancreas. And if you look at this model, this is a RAS P53 mutant with only targeting the pancreas. And you can see this incredibly large pancreas tumor here. The mouse is very sick. Within 10 weeks, there's a lot of ascites. And when you knock out MSASI, what you see is a much smaller tumor. And this is just showing you the volume drop that occurs. But I think what's really interesting is that if you look histologically, this is just a normal pancreas. If you look at the tumors in the wild type, it's all sheets of adenocarcinoma. If you look at the knockout here, I'm showing you MSASI 2 knockouts. What you see is that there's a lot of like normal tissue still left, and the aberrant tissue that's arising are few. And what was really important is we worked with the pathologist who actually diagnoses pancreatic cancer to ask what the ratio was between early lesions like Pennins relative to adenocarcinoma. So what he found with us is that if you look at the wild type, everything is adenocarcinoma. This is what drives lethality. But when you look at the MSASI knockout, most of the lesions that are arising here are PANINs or pancreatic intraepithelial neoplasias 1, 2, and 3. These are all sort of pre-malignancies essentially, and not an end-stage malignancy. So what this is saying is that when you delete a stem cell program like Masashi, it essentially disables the power of oncogenes like RAS and tumor suppressor losses like P53 from taking a pre-malignant lesion into a full-on malignancy. And I think that says a lot about the importance of stem cell programs in enabling oncogenic growth and oncogenic targeting of normal and pre-malignant tissues. So for a long time, after all that work, I've been interested in trying to develop a Masashi antagonist. And we have done many things over the last few years, including currently. But one of the things we did with Ionis Pharmaceuticals is really develop the antisense oligonucleotide, because essentially there's like an RNA binding a protein, and we thought this might be a good way to get, more effective way to get at targeting Masashi. So Ionis had generated multiple ASOs for us. We tested about 20 of them. They generated 400, and of the 20, we picked 2 that were effective at running down Masashi levels, and this shows you colony area reduction, and that also when you deliver it in vivo in intratumorally, because this was not lead optimized at the time, you see reduced tumor growth and you see a reversal of the rate. So this is really saying if we were able to develop an antagonist, it could very much have an opportunity to turn down the disease and to control it better. So along with trying to turn it on and off, we've also been interested in understanding really the contribution of Masashi positive cells to disease and to normal growth. So to this end, we developed a couple of reporter mice that started at Duke, and this shows you that we have knocked in YFP into the Masashi1 locus endogenous. So it's an endogenous reporter, GFP into the endogenous locus of Masashi2. And these mice, for example, have bright green eyes because their retinas are positive. Masashi is expressed there, as well as they are enriched in areas where there are progenitor cells and stem cells, hippocampus and SVZ in the brain, and in the bone marrow, stem cells are high and differentiated cells are very low. So although reporters, come with their own set of problems, this really mimicked Masashi expression quite well. You can do a lot of things with this, and we hope that this is an asset to the community to look at stem cells across many tissues and also its role in cancer. What we really did was cross this reporter, so reporter Masashi 1 or 2, REM1 or 2, into the KPC mouse model. And what that did was that really illuminated the heterogeneity that exists within the cancer. So when you just go look, everything is RAS and P53 mutant, but when you start looking with the lens of Masashi, you see that some fraction of those cells are actually Masashi positive, and the rest are Masashi negative, and this is 90% versus 10%. What was interesting is that if you transplant them in equal numbers, the reporter positive is more aggressive, they make organoids and the reporter negative don't, and they drive all the lethality in this very lethal cancer, and the Masashi negative cells really don't. Perhaps the most interesting thing out of all of this was that when we treat these mice with gemcitabine, which is standard of care, although not as effective as all of us would like it to be, if you look at vehicle control, about 7 to 10% of the mice are positive for Masashi expression in a normal tumor. But as gemcitabine drops in, the reporter negative cells are depleted and the reporter positive cells accumulate. And so what this is saying is that the most drug-resistant population in pancreatic cancer are really the Masashi reporter positive cells, or Masashi positive cells, and it endows these cells with an aggressive and drug-resistant state. And this is just to show you that it is not only in pancreas cancer. We've used this model now to look and probe heme malignancies like CML and blast crisis CML, and you can see here that Gleevec, which if it works really well in chronic phase and not so well in blast crisis, for example, is very effective against the reporter negative cells in leukemia, and the reporter positive cells, which are Masashi positive, act like there is nothing going on. So completely resistant to this drug as well as other sources of stress like radiation. So I'll just take a couple of minutes and go into how we've used this in context of imaging. So we are very interested in understanding how all of this knowledge that our community generates about cancers, molecular, biochemical, cellular, how it all really like works in vivo and how it sort of collectively drives this very lethal disease. And so we've worked over the last few years to really develop imaging strategies so that we can non-invasively understand how cancers grow in vivo. So here, for example, I'm showing you GFP positive cells from leukemia cells that are transplanted into a backlit recipient. So the body is obviously dark, but if you put in red, all of the cells can be lit up. And this is an example for to show you that in the bone marrow, there's a blue mouse, a cyan mouse, lit up on the marrow, and the edges of the bone are lit up with a notch reporter, and we've transplanted in red hematopoietic cells. And you can see that we can see these moving around with a lot of high resolution, spatial, and temporal clarity. We've used this to also then mark up subdomains within the microenvironment, like we've used anti-V cadherin to mark up the blood vessels. And we published several papers on trying to understand where hematopoietic stem cells normally reside and connect with. But I just wanted to, what I want to show you is how you can use this in cancer. So what we've done here is use a backlit red mouse, again, non-invasively, transplanted with leukemia cells, all in green, blast crisis CML. And what you can see is that although leukemia is thought to be this liquid tumor, it's very much not liquid in the middle of the microenvironment of the bone marrow. So all of these cells, there are some cells that are moving around, obviously, but many cells are entrenched in the marrow, interacting with the microenvironment and blood vessels for hours at a time. And we have shown in data that I can't really share because of time, that this really allows leukemia stem cells and cancer stem cells to form bridges between the leukemia and the microenvironment. And these adhesion molecules like CD98 tetraspanin and syntecan-1 in papers that we published recently are really critical to support the growth and survival of these cancer stem cells. This is also showing that you can switch this around and pivot to looking at Musashi positive cells within the leukemia. Here the leukemia is red and the reporters are green, so that are the minority. But when you treat with a drug, the reporter positive cells become sort of dominant. And then you can use this to really understand whether reporter positive cells and Musashi cells really lie in particular areas or niches where they're protected from being targeted by standard of care. We've also moved this into pancreas cancer. So this is just actually a normal pancreas, and this is a pancreas tumor. So what I'm showing you here, the blue is all of pancreas cancer, all RAS P53. Imagine a very large sheet. The pink are all blood vessels here, and the green are the Musashi positive cells. And it's just really interesting that they're in these clusters across. I thought they would be disseminated, but without the sort of live imaging, I think we would never have understood that they're really in these subdomains within the cancer. And we're really interested in understanding whether there's a particular microenvironment that cloaks it to protect it from these therapies and give it sort of that aggressiveness. So beyond looking at the microenvironment, which is work that we're just starting, I'll just show you some of the last pieces of data, which is also that we've worked a lot to understand the molecular infrastructure of these Musashi positive cells, why they're so aggressive and why they're so therapy resistant. And so what we've done is use this Musashi reporter KPC mouse to sort out reporter positive and reporter negative cells, and done genome-wide RNA-seq, ChIP-seq, as well as the genome-wide CRISPR screen in 2D and 3D conditions. And what this shows you, I've already told you that the reporter positive cells are more aggressive. This shows you molecularly that they're different from the reporter negative. They have particular kinds of signatures like development and stem cell signatures, and they're also high for cancer relapse signatures, which is aligned with the fact that they don't go away when you deliver drugs. So if they stay around, they will be the ones that trigger a relapse, and that's aligned with this. And this is just to say that we also looked at stem cell super enhancers with ChIP-seq, and you can see that Musashi 2 is a super enhancer, much to my joy. and in non-stem cells, it is not. So one of the things that differentiated our work from others that were also doing CRISPR at the time is that we did everything in primary cells. So we used KPC cells and we did 3 independent lines. 100 million cells went into this. So it was like a real labor of love by my student, Nikki Lytle. And we did a 2D assessment first, but we then did an analysis in 3D to look at what genes were necessary or dropped out in 3D. And the reason we did this is because only the Masashi positive cells will grow in 3 dimensions. And so it's a very, it's an intelligent sort of way of focusing in on genes that are really important for stem cell growth. And then separately, we've done a synthetic lethal with gemcitabine. So we've integrated the CRISPR RNA-seq data to identify genes that are enriched in and essential for growth of stem cells. And this allowed us to really develop, through string interactome, a network map of the key genes that are important in pancreatic cancer stem cells. So this is just showing you like a 30,000 foot view of all of the genes that came out. So they're now sort of coalesced into programs like developmental pathways, cell adhesion, and so on. What we did was we really focused on things that we didn't expect to see, like immune pathways and cytokine signaling, as being important in the epithelial cancer cells. And we subsequently focused on things that were actionable and maybe targeted, and particularly RR gamma. And RR gamma has been shown to be important in IL-17 and Th17 growth. And because of that, a lot of companies have made RR gamma antagonists. So what we did was we tested to see if RR gamma could be blocked and may be effective against pancreas cancer. So this is just showing you 3 independent shRNAs, growth in vitro and inhibiting growth in vivo. And then we also use the inhibitor, this is SR2211, but we've also used actually clinical agents to show that you can run down effectively KPC sphere formation, as well as increase survival in autochthonous KPC models. So this is just sort of a summary saying that a lot of these pathways that we think of in the microenvironment and in the immune system, what we are seeing with this CRISPR strategy is that a lot of these are actually being used directly by the epithelial cancer cells themselves, and particularly the stem cells. This includes IL-10, IL-34, IL-17, which I didn't talk about, as well as ROR gamma. So what we feel is that these cells may have co-opted the immunomodulatory microenvironment, and it selects for cells that can grow and essentially utilize it for their growth. But it also offers an opportunity to really reposition immunomodulatory agents in pancreatic and other cancers, because so many of these have been targeted in the immune system and in autoimmunity, that one of our goals through Stand Up to Cancer is really to try to reposition this for cancers that very much need it. And this is just sort of a point I like to make, that just a thing to keep in mind, that obviously everyone wants to activate the immune system against cancer, but activating it sort of without understanding the complexity may really trigger regrowth of tumor cells. And in that context, some clinical trials have shown sort of aggressive growth for some patients. And I'm just going to end with a new model that we've made, and I'm not going to say a lot about it because it's not finished, but the last thing we've done is made a Masashi2 pre-RT2 mouse to really understand whether cancers can start from Masashi positive cells. So we've made like knockouts, including Dr. Akano's knockout that we've worked with. We've made reporter mice that have told us a little bit about how these cells function. And now we've made a pre-ER mouse that will only allow deletions in Masashi positive cells to ask whether cancers can start in these cells. And this shows you that when we cross into a MIC mouse and use tamoxifen, we get cancers across the body. And when we cross it into another mutation like RAS, we get a completely different set of tumors. And interestingly, in the pancreas, what we've been really chasing down is that it makes multiple subtypes of pancreatic tumors. So people study pancreatic ductal tumors a lot, but adenosquamous carcinoma is actually the most aggressive subtype of pancreas cancer, and acinar cell carcinoma is enriched in pediatrics. And these are not usually modeled, and this Musashi CRIAR allowed us to really model this, and we're using single cell sequencing to really understand the steps that a completely normal cell takes to become a pre-malignant cell, and then diverge along different paths to make different tumors. So I'm just going to end here and thank all of the people who actually do the work, Nikki Lytle and Ray Fox, who really did all of the pancreas work initially in our lab, together with Andy Lowy, who's like a real critical partner for us, and many others like Nirakar and Mike, who helped generate this new mouse, the Musashi CRIAR, and that we're following up, trying to understand where cancers begin and how they grow. A lot of the leukemia work was done by Kyle and Jivisha Bajaj, and I would be remiss to not thank Dr. Okano again. He's been a great collaborator, partner, inspiration. You heard Musashi throughout the talk, so it's really a pleasure to be able to talk here, and also have included his data and have the opportunity to collaborate with him directly. So I'll stop here.
Thank you very much, Professor Raya, for a beautiful talk. We have time for questions if you'd like to post your questions in the Q&A window. And seeing none, maybe I will start by asking one. I'm sure Akana has many. So given the link between Musashi signaling and immune signaling, do you think you could sort of design a modified CAR-T type of therapy that might avoid those immune pathways or even suppress them while activating others?
Yeah, so we've kind of looked at a lot of downstream targets of Musashi in an attempt to target the pathway. And one of the things that we have identified and published on is a tetraspan and tetraspan 3. And it's very much under the radar because we found it as a target of Musashi, and then we went ahead and made a knockout and had an impact in heme malignancy. And then we were funded to make an antibody therapeutic against it, so which we've done. And so we've kind of considered making that into a CAR-T possibly. So we are like around Musashi looking at what else Musashi is really controlling, because in addition to then targeting Musashi, we may be able to target other pathways that are in the immune sort of collective, if you will, and then try to use that as a strategy to target these very resistant populations.
Might be interesting to collaborate with someone like Pradipta Ghosh to see if we could build network models to sort of try and predict the behavior of the system, given these interesting subsystem interactions.
Yeah, that would be great. Thank you.
Professor Kanner, do you have any questions?
Yeah, thank you very much. I just jumped to Salk Institute. Okay, so so thank you very much for showing some of our collaborative research. And so because it's a very nice example of the UC San Diego and the Keio collaboration, and also in addition, Ionis, a strong biopharmaceutical venture company in San Diego, is collaborators. It's a very nice collaboration between the academia and so venture company. Okay, let me make some more scientific question. And so as you have performed gecko screening and so identify some potential downstream events of the to the Musashis, and also in addition, as you know, we are performing the direct the downstream by performing hit script analysis, binding partner as messenger RNA binding partner to the Musashis. Okay, we are now interested to compare each other, which is a direct target and indirect. Mm-hmm. So it's interesting to see that Musashi rarely bind to the immunoregulatory or the inflammation regulatory molecules or not. So we take a look at our the data again, and so we make a feedback to you. Okay.
That would be great, actually. Yeah, I think that's a really good point, because obviously we were sort of looking programmatically, but I think it'd be really interesting to see what is directly bound by Musashi and what is sort of like a secondary event. So I would really welcome that collaboration. And again, thank you for like all of the work that you did that led to this work.
Okay, and also I'd like to ask, even the young people and high school students to make, even if it's the fundamental or basic questions. Is it okay? Do you accept those, questions?
Of
Right?
course. Yeah.
はい、高校生の皆さんどうぞ、本当に基本的な質問でもいいですから、Q&A に質問を送ってください。後で、来てますかね。Okay, there's some questions.
There is one question in the Q&A from Masaki Ishikawa.
Yeah.
And basically asking about this need for tumor targeting of MSI because of MSI functions in other tissues like neurons. So is that a problem?
Yeah, so, it's definitely a concern. I would say Musashi is really critical embryonically and in development more than in the adult. And hedgehog signaling, I think, was a really good example where there was the similar worry, but ultimately it was actually quite non-toxic, there was a little bit of tox, but very much tolerable. And what we've done, and you have to also compare it to what we're using currently, like chemo, radiation, things that are incredibly tough on the rest of your body, and we basically try to find a therapeutic window. Nonetheless, I think it's a very reasonable and a good question. And so what we've done is we've made the Cree-ER, and then we've conditionally knocked it out in the adult in an effort to understand what tox we might see if we are delivering a drug. Should we successfully make a drug and deliver it? What other kinds of tox compromises we might see? So we have already done that for Musashi 1 and do not see any overt defects. And we're in the middle of doing it. We've done them both individually, and we're in the middle of trying to do it combinatorially. And I think those questions, ultimately, we may not know the answer until we do the empirically a clinical trial. But I think that's our hope, is that there's enough of a window there that although developmentally it's so critical, that it may be tolerable, or the tox may be tolerable in an adult context. But that's a great question.
Yeah, please let me make one more comment. This Ishikawa-san's comment is very nice comment. Actually, the Ion is developing antisense oligo to the Musashis, okay, as Musashi 1 and Musashi 2. Now, recently, there's a rapid progress of the drug delivery system antisense oligo, even to the brain, central nervous system, and particular organs. So that will be one of the method to address the specificity issues. That we hope that Ionis is very rapidly working on it. Yeah.
Yes, that's true. That's a good point.
Okay, well, so maybe it's due to time to switch next speaker. Okay.
Yes. Thank you very much.
Thank you very much.
For a wonderful talk.
Thank you.
And for sharing these new and really exciting results. So
thank you.
Thank you.
Let me hand over to Professor Kano now.
Okay, so it's my great pleasure. I can introduce Professor Hideyuki Saya, one of my colleagues in Keio University School of Medicine. We have been friends and collaborators over 20 years, and he did a very great work last week. He worked as the president of the annual meeting of the Japanese Cancer Society in Pacifico, Yokohama. This meeting was rarely done, Yokohama. Okay, we met in-person meeting for a long time. The social isolation. It is very fresh feeling for me, and so I like it. And so Professor Saya has been working cancer stem cell for many years, and we've been talking new therapeutic method targeting the cancer stem cells. Okay, Dr. Saya, please.
Okay, thank you very much, Dr. Okano, for your kind introduction. Let me share my slides to you. Okay, just a moment.
Yes, please start slideshow.
Can you see my slide right now?
Yes.
Okay,
yes.
So let me start. Okay, so.
Yeah. Yes. Okay. Great.
Just a moment. Okay. So I'd like to express my great appreciation to the organizers for giving me a wonderful opportunity to talk about our cancer stem cell research in next 25 minutes. Okay. So actually, cancer stem cells are subfraction of the tumor cell, which have a highly tumorigenic activity and also resistant to the various therapeutic intervention. When you treat the cancer tissues by the chemo and the radiotherapy and the molecular targeting therapy, cancer stem cells remains. So therefore, a tumor sooner or later back again. Therefore, we really need to destroy the cancer stem cell so as to completely eradicate the tumors. Okay. So therapeutic resistant mechanism of the cancer stem cell has been extensively analyzed. 4 major mechanisms as shown in this slide: slow growth and quiescent. And also the cancer stem cell has a high drug efflux and the detoxification by using the ABC transporter and also aldehyde dehydrogenase. Also, cancer stem cell has a characteristic to suppress the immunologic reactions by the expressing the PD-L1 or other cytokines such as IL-10. But we have special interest in redox regulation mechanism of the cancer stem cells. So cancer cells are exposed to the high level of the oxidative stress, both intracellularly and extracellularly. The hypoxia and the hypoglycemia are a major cause of the ROS induction. Proliferating cells compete to obtain the oxygen and the glucose, leading to the hypoxic and the hypoglycemic environment in the tumor tissues. Furthermore, the oncogene activation and the mitochondrial mutation, even a treatment that increase the level of the ROS. So therefore, the ROS is always all around the cancer cells. Cancer cells in hydrogen peroxide is a change to the hydroxy radicals in the presence of the Fe ions. A major target of the hydroxy radical is the lipid and generating the lipid ROS. Okay. Lipid ROS interferes the composition of the cell membrane, resulting in the non-apoptotic and oxidative cell death, oxidative induced cell death. That's what we call the ferrotosis. Okay. The cancer stem cell has a high ability to suppress the ROS and consequently avoid the ferrotosis. So CD44 is a major cancer stem cell marker for the various epithelial cancers, such as the breast and the prostate and the head and neck and the colon and the pancreas and the gastric cancers. But we really want to know the CD44, the just marker for the cancer stem cells, or CD44, there's something to do with the cancer stem cell functions. CD44 is a transmembrane protein and also finds to interact with lots of extracellular matrix. But at CD44, somehow the expression has been implicated in a tumor growth and invasion and metastasis. About 30 years ago, my laboratory identified CD44 as an alternative splicing variant. So actually, the CD44 has a 10 variant exons in the extracellular domain, but particularly V8, 9, and 10, these 3 exons are often inserted in CD44 extracellular domain in the cancer cells. But at that time, we really don't know the significance of this variant CD44 expression in the cancer cells. But about 10 years ago, our laboratory identified the function of this variant CD44. CD44 V810, this variant form of the CD44 expressed on the cell membrane to interact with the XCT cysteine transporter to stabilize the expression of the XCT cysteine transporter, which allow the cell to incorporate the cysteine in the cell. The cysteine was used for the synthesis of the glutathione. As you know, the glutathione is a major antioxidant. So therefore, a CD44 variant expressing cell can reduce the reactive oxygen species level, leading to the resistance to the chemo and the radiotherapy. This imaging mass spectrometry shows the CD44 variant expressing cell, which is metastasized into the lung, has a high level of the reduced form of the glutathione. But you can see the oxidized form of the glutathione is not high level in a CD44 variant expressing cell. So we really want to know we are wondering which is the better treatment target, whether XCT or CD44 variant. Given that we found that XCT is stabilized on the cell membrane by interaction with the EGFR in the glioma cells, which was not expressed the CD44 variant. So therefore, we decided to select the XCT as a common target for the cancer stem cell. If we can block the XCT expression or XCT function, we may be able to reduce the glutathione expression, which increase the reactive oxygen species level in the cancer stem cells. Okay. With screening the FDA approved drug library, and finally we found sulfasalazine, which is the old drug used for the ulcerative colitis and the rheumatoid arthritis. This sulfasalazine can suppress the function of the XCT. This is the xenograft model of the human head and neck cancers. As you can see, the sulfasalazine treatment of the mice can effectively reduce the expression of the CD44 variant. So as you can see in the flow cytometry, the sulfasalazine selectively eliminate the CD44 variant expressing cancer stem cell fractions. Okay. In order to know the role of the CD44 variant and XCT complex in a tumorigenesis, we use the transgenic mice developing the spontaneous gastric cancer by co-expression of the Wnt1 and the prostaglandin E2 in the gastric mucosal cells. These mice were called in a GAM mice, gastric neoplasia mouse, and developed by the Dr. Oshima's group in Kanagawa University. It is the very interesting mice because all mice developed a well-differentiated adenocarcinoma within 30 weeks after birth. Okay, we cross this mice with a CD44 knockout mice, and resulting in tumorigenesis completely suppressed by the knockout of the CD44 molecule. Okay. So therefore, we started to treat these GAM mice by using the sulfasalazine to suppress the XCT function 10 weeks after the birth. Okay. Control mice developed the huge, well-differentiated adenocarcinoma in the stomach within 30 weeks. However, sulfasalazine-treated mice obviously suppressed the tumorigenesis in the GAM mice. And also the Ki67 in a proliferation marker is obviously low expression in a sulfasalazine-treated mice. So now I summarized our basic experiment up to here. CD44 variant and XCT complex protect the cell from the high level of the reactive oxygen species. and the CD44 variant and XCT positive cells contain the functional cancer stem cells in epithelial tumors. And the XCT inhibitor sulfasalazine specifically suppress the CD44 variant positive cell survival, and also sulfasalazine significantly block the tumor genesis in a mouse experiment, and also metastasis, which has not been shown today. So therefore, we actually decided to go into the clinical application using the sulfasalazine. First clinical trial, we used the sulfasalazine monotherapy for the advanced gastric cancer patient, because we know the gastric cancer contain the CD44 variant positive stem cell fraction in more than 70% of the cases. Before and after the sulfasalazine treatment, we performed the biopsy. Okay, as you can see, the pre-treatment of the sulfasalazine, we can see the CD44 variant positive population, but 2 weeks after the treatment, we can significantly reduce the CD44 variant positive cells. So therefore, we could obtain the proof of concept in those clinical trial. Okay. So then we'll proceed to the next clinical trial by using the patient who has advanced inoperative lung cancer by using the chemotherapy and the sulfasalazine. But those patients was treated by the conventional chemotherapy agent, cisplatin and the pemetrexed and sulfasalazine oral treatment. Result was very clear. Usually, those inoperative lung cancer patient has a recurrent tumor within 4.3 months. So therefore, median progression-free survival is very short. However, our effort, addition of the sulfasalazine, markedly extend the progression-free survival to the 11.7 months. So actually, the conventional chemotherapy agent may kill the non-stem cell population of the cancer cells, and the cancer stem cell population may be suppressed by the sulfasalazine treatment. So sulfasalazine treatment has a pros and cons. Advantage of the sulfasalazine is that it inhibits the XCT. The XCT specifically blocks the glutathione synthesis in the cancer stem cells and the resistant cells. Okay, usually normal cells and the normal type of the cancer cell does not use the XCT to incorporate the cysteine to synthesize the glutathione, because the glutathione can be synthesis with the serine and the methionine inside the cells. So therefore, XCT inhibition is specifically works for the cancer stem cell and the resistant cell. But a disadvantage is only limited fraction of the tumor cells are sensitive to the sulfasalazine. Also, our experiment shows in a sulfasalazine sensitive cell become resistant during the course of the treatment with the sulfasalazine because of the reason to reduce the level of the reactive oxygen species. So therefore, we really want to find any way to increase the sensitivity of the cancer cell to the sulfasalazine. Then we performed the screening of the synthetic lethal screening. So synthetic lethal screening, we have a sulfasalazine resistant cell, HSC4. This cell was treated with sulfasalazine plus one another FDA-approved drug. We actually screened around about 1,200 FDA-approved drug. As you can see, most of the drug actually, those cells, the effect of the drug is not changed with or without the sulfasalazine. But only one drug right here, okay, is very effective when we combine to use the sulfasalazine. So that drug called the dichlorine, okay. Combination of the diclonine with sulfasalazine will dramatically enhance the effect of the sulfasalazine in this resistant cells. So therefore, we identify the drug which can induce the cell death only in the presence of the sulfasalazine. Well, diclonine can sensitize the HSC4 cells to the sulfasalazine. But diclonine is a local anesthetic drug. So therefore, we can't use this drug for the patient, okay? However, diclonine has a structure as a covalent inhibitor of the aldehyde dehydrogenase. So you can see the aromatic ketone and the amine structure, which is specific for the ALDH inhibitor. Therefore, we try to find some of the oral taking drugs which have a very similar structure to the diclonine. And then we found it. We found the oxyfedolin, which is a very old vasodilation drug, has an aromatic ketone and amine structure. We combine to use the oxyfedolin with the sulfasalazine. This combination is markedly kill the resistant cancer cell, such as the HCT116 and the SCC4. Combination of the sulfasalazine and the oxyfedolin is very effective to kill the resistant cells. What is the mechanism? The lipid loss, high level of the lipid loss is quickly converted into the aldehyde called 4-HNE. Aldehyde 4-HNE is very, very toxic in the cells. However, most of the cancer stem cells or resistant cell, the high level of the aldehyde dehydrogenase to degrade the 4-HNE. So therefore, cell can survive. We use the oxyfedolin or diclonine to block the aldehyde dehydrogenase, which leading to the very high level of the 4-HNE to killing the cells. But we can confirm the 4-HNE expression started to be very high when we combine to use the oxyfedolin and the sulfasalazine. And also the one more evidence, we analyze the profile of the expression profile of the sulfasalazine resistant cell and the sulfasalazine sensitive cell. As you can see, the ALDH3A1 is very, very increased in sulfasalazine resistant cells, okay? So therefore, we perform the siRNA experiment to reduce the ALDH3A1 level, and that this ALDH3A1 depleted cell become very sensitive to the sulfasalazine, okay? We tested many different cell lines. Sulfasalazine alone and the oxyfedolin alone did not have so much effect in those cancer cells. However, when we combine to use the sulfasalazine and oxyfedolin, as you can see, most of the cells become sensitive to this combination therapy. We performed the in vivo experiment. HCT116 is oxaliplatin resistant. Oxaliplatin is a very, very strong anti-cancer chemotherapeutic agent, but this colorectal cancer cell is resistant to the oxaliplatin. The xenograft of the HCT116 cell was treated with the sulfasalazine and oxyfedolin. As you can see, this combination therapy completely suppressed the tumor growth of the HCT116 cells in vivo. Then, what is the mechanism? Okay, so XCT is suppressed by the sulfasalazine treatment. That's reduce the level of the glutathione. Okay, that then they increase the level of the ROS. Okay, ROS was converted to lipid ROS, and the lipid ROS is converted to 4-HNE aldehyde. But in a resistant cell has a high level of the aldehyde, which can degrade the 4-HNE. But if we use the oxyfedolin, can block the escape route of the cancer cells, which increase the expression of the 4-HNE. So 4-HNE aldehyde can covalently bind to the cysteine, which reduces the activity of the glutathione again, and also the other reducing molecule, which increase the more high level of the ROS. Okay, ROS converted to the 4-HNE. So therefore, this vicious cycle for the cancer cells leading to the cell death. We are now actually planning to the clinical application of the combination of the SSD and oxyfedolin. Also, radiation therapy can increase the high level of the ROS. So therefore, we are also planning to the combination of the radiation plus oxyfedolin and the radio chemotherapy plus oxyfedolin. So I'm going to change my gear to the next characteristic of the cancer stem cell, slow growth and quiescent. Okay. To address this issue, we use the bone marrow stroma cells collected from the INK4 ARF knockout mice. Okay. This bone marrow stroma cell has a mesenchymal stem cell-like characteristic to be differentiated into the adipocyte, osteocyte, and the chondrocyte. This BMSC was introduced with the MYC oncogene with the GFP, and the GFP positive cell was inoculated back to the syngeneic mice. The mice is died within 60 days by development of the malignant tumors. The tumor histology shows the osteosarcoma, but this heterogeneous tissues has a osteogenic differentiated part and a chondrogenic differentiated part, and also very immature tumor cells. But these tumors, it metastasized into the lung and liver very quickly. Okay. To further characterize this osteosarcoma cell, we performed the single cell cloning. Okay. Then we found this osteosarcoma tumors were composed by the two different populations of the cells. One is the AX cells. AX cells had a bilineage differentiation characteristic and also very high tumorigenic and rapid growing cells. As you can see, the X cell can be differentiated in osteocyte and the chondrocyte, but not in adipocyte. However, AO cells can be differentiated in the three lineage, adipocyte, osteocyte, and chondrocyte, like mesenchymal stem cells. Okay, but AO cells is a low tumorigenic and slow growing, but eventually converted to the X-like highly proliferated cells. So difference between the AO and the X cell in adipogenic differentiation. So therefore, PPAR gamma is a master regulator for the adipogenic differentiation. The AO cell had a PPAR gamma positive and the AX cell is PPAR gamma negative. We compare the sensitivity of the drug sensitivity of these cells. As you can see, the high tumorigenic AX cells is much more sensitive to the anti-cancer drug. So therefore, when we mix the AX green cells and AO red cells treated by the adriamycin, as you can see, the red AO cell is much more resistant, and the AX cell will sensitive to the chemotherapeutic agent. When we back to the original osteosarcoma cells, the most of the cell is an AX-like cells, but a small number of the PPAR gamma positive population in the AO-like population. When we treat this osteosarcoma by the adriamycin, which is the anti-cancer drug, most of the cell remain as a PPAR gamma positive population. So therefore, AO population is much more chemoresistant. Okay. So therefore, when the osteosarcoma tissue was treated by the chemotherapeutic agent, the most of the AX cells were disappeared by the drug treatment. However, AO cells remained, and eventually AO cells turns to be AX-like cells. Okay, and then AX cells were recurrent of the tumors. Recently, our laboratory identified actin dynamics regulate the differentiation of the mesenchymal cells. Okay? Usually, mesenchymal cell has a MKL1, which is the transcriptional regulator inside the nucleus to suppress the PPA gamma expression. But when the actin cytoskeleton is depolymerized by the ROCK inhibition, and this monomeric actin was interact with the MKL1. MKL1 is a translocate from the nucleus to the cytoplasm, which allows the cell to express the PPA gamma, and cell was differentiated in the adipocyte. So therefore, by controlling the actin cytoskeleton, we can induce the differentiation to the adipocyte. Okay, so this is the AL cells were treated by the ROCK inhibitor Y compound. Clearly, we can induce the mature in adipocyte differentiation. But actually, Y compound is a very, very strong chemical. So therefore, we try to find an FDA-approved drug which have a ROCK inhibitory activity. Fortunately, we found a Fasgil, which is a drug for the vasospasm in a subarachnoid hemorrhage, has a ROCK inhibitory activity. And Fasgil can clearly increase the adipocyte-specific gene expression and induce the adipocyte differentiation in AL cells. So therefore, we try to treat the osteosarcoma tissue by the chemotherapeutic agent to kill the AL-like cells, and then subsequently treat those tissue by the ROCK inhibitor or MKL inhibitor to induce the differentiation of the AO-like cell to the matured adipocyte. So we tried this experiment in vivo model system. The osteosarcoma cells were inoculated into the mice and treated with adriamycin alone, Fasgil alone, and the combination of the adriamycin and the Fasgil. Clearly, this combination can suppress the tumor growth, and also the immunocytochemical analysis shows in a combination therapy is significantly reduce the tumor size and also in adipocyte differentiation. Okay. Finally, shows the summary of the second part of my talk. In osteosarcoma, is composed by the two different types of the cells, AO and AX cells. AO seems like a cancer stem cell population. AX cell easily to kill the conventional chemotherapy, but AO will be remained. So therefore, AO cell will be induced the differentiation to the mature adipocyte by the ROCK inhibitor Fasgil treatment. So in a cancer, tissue is composed by the heterogeneous populations. So it's very important to characterize those heterogeneous components. But a heterogeneous component usually generates not only by the genetic mutation, but also the non-genetic events. Usually, those non-genetic events is based on the plasticity of the cancer stem cells. For the treatment of the heterogeneous cancer, we need to use the drug which can targeting the different component of the tumors. So I would like to acknowledge the people who have done all the experiment I have talked today. Mainly, the first part of my talk is done by the Dr. Nagano's group, and the second part of my talk was done by the Dr. Shimizu and Dr. Nobusue's group. Thank you very much for your attention.
Thank you, Professor Saya. Very thoughtful and very informative talk. Okay, now paper is open for discussions. Actually, so you have mentioned about chemosensitive and chemoresistant cells. They are very important component of the very heterogeneous cancer tissues. So Dr. Saya has developed very selective therapy for each population. This is works for the cancer stem cell and non-cancer stem cells. This is very important point, I believe. Anyway, so I would just young people, including high school students, may question, even if it's very fundamental, primitive, whatever. Any questions? Anyway, so I forgot to mention about Dr. Saya. He's a pioneer of the drug repositioning using the FDA-approved drug library. Actually, so in collaboration with Saya, we have developed a new anti-ALS candidate drug, okay, with his library and developing laws, and we have finished clinical trial. I believe it worked, okay, so his method is very great. Anyway, so any question? Tanishita, do you have any question, comment?
Yeah, I was, can you guys hear me?
Yes.
Yeah,
Yes.
it was really impressive work. I mean, I had a question basically about the osteosarcoma data where you were like differentiating it. I wasn't sure, are you doing that in vivo or where
In
is
vivo.
that being done?
Yes, in vivo.
Okay.
The final, because I do not have so much time to explain the experiment, but the final part of my talk is an in vivo experiment. Osteosarcoma model was treated by the conventional anti-cancer drug with the ROCK inhibitor, which can treat it. Yes.
I thought that was great. So are you seeing sort of sustained, like sometimes, you know, people will ask like, how long do you have to do this and how durable is the remission? So what is your feeling about it? Like, how long are you treating? How durable are the remissions? Can you comment on that?
Yeah, that's great, question. Because the ROCK inhibitor, we need to be treat those tumors by the ROCK inhibitor for a long time, because otherwise, all the differentiated adipocyte will be back to the more primitive cell again. So therefore, at least we need to treat those tumors in a longer time. It may be more than a couple of weeks. So that might be one of the very important point, okay? So, because if I actually quit the treatment of those cells, maybe I'm actually concerned about the adipocyte differentiated cell to back to the primitive cancer stem cells again. Okay.
Is okay?
So you might need like a sustained treatment, basically.
Yeah, definitely.
It's great.
Yes.
It looks really impressive. Thank you
very much.
Okay, thank you. Okay, there are two questions in the Q&A box. One is from Masaki Ishikawa, and the same person, okay, who did the previous question. Okay, thank you. I understand the cancer cells are very heterogeneous. In the second part, osteosarcoma project, why you don't target loss as well in the first part? Okay, very important. Yeah.
Yes, yes, that's right.
Yeah.
Actually, it's a little complicated, and I actually extend the first part. Most of the cancers are caused based on the inflammatory reaction. Those cancer stem cells are caused by the inflammatory reactions. Those cells has a very high level of the reactive oxygen species. However, the cancer stem cells caused by the genetic mutation, like a MYC or RAS or those type of the genetic mutation, do not increase so much level of the ROS. Okay, so therefore, I think the cause of the different cause of the cancer stem cell that we need to select the different treatment. Okay.
Okay, so there's another question in Japanese, and please let me translate. Okay, so the investigation using model animal appears promising at a first look. Okay, if you apply to the human, there should be some species differences in terms of the efficacy and so safety issues. How do you think about it?
Yeah, definitely, that's the concern. However, we cannot we cannot do it more in a model system. So therefore, we need to do the clinical trial. Okay, definitely clinical trial will be started from the very low dose. Okay, on the ROCK inhibitor and also the anti-cancer drug to confirm the safetiness, and then we would like to see the effect of those drugs. So therefore, the clinical trial will necessary to confirm our hypothesis, our model animals experiment is right or not.
Actually, so please let me comment about second question. So I believe that to bridge model animals and human clinicals, I believe the human iPS in vitro model is a nice intermediate step, So in our case, in the ALS, we didn't use animal model. We just use a human ALS patient-derived iPS cells, and it worked in the clinics. Okay, it is one of the methods. If we combine with the drug repositioning, it's a very wonderful system. Anyway, is there any questions? Yeah, today there are lots of questions. Very, very exciting presentation was there. Okay, thank you very much. I'll back to Soyama-san and Waga-san. Thank you very much for the speakers.
Thank you so much.
Soyama-san, Waga-san, どうぞ。
Thank you very much for the wonderful presentations and the interesting discussion, the Q&A session. The COVID-19 pandemic has taught us the importance of long-term investment in science and technology, not just to explore solutions to difficult problems in society, but also respond to global crises at unprecedented speed. And building and nurturing an international network of relationships in basic and applied research, medicine, and education seems more important than ever before. So I'd like to encourage the audience to stay in touch with us at UC San Diego and Keio University to find out new discoveries and breakthroughs in the field of regenerative medicine. If you have any questions, please send us messages. We can stay in touch. And I want to thank LinkJ leaders and staff for their generous support for this joint webinar series. And with that, let's have Soyama-san for his closing remarks.
はい、ありがとうございます。まずはですね、ご登壇いただきました谷下先生、さや先生、マクロ先生、岡野先生、和賀先生、誠にありがとうございました。そしてまたですね、朝早い時間からご視聴いただきました皆様、本当にありがとうございます。特にね、高校生の皆様は今回ね、多数参加されています。いろいろとね、難しいところもあったかもしれませんが、ぜひですね、最先端のこういった、科学のディスカッション、に触れていただいて、興味をですね、もっと持っていただけたらいいなというふうに思っております。リンク J ではですね、このウェビナーシリーズも含めてですね、いろいろなウェビナーやってまして、その中にはですね、リンク J のホームページ等にですね、アーカイブしているものもございます。例えばですね、この領域、ライフサイエンスの領域のスタートアップの皆さんの具体的な取り組みですとか、あとは政策キーパーソン、規制とか女性がいっぱいある、とっても大事な、命に関わる分野だから、そういうものがいっぱいあるんですけれども、そういった、政策キーパーソンの方が本音で語るみたいな、そんなシリーズのアーカイブもありますので、ぜひですね、ご関心のある方は見ていただいて、よりヘルスケア、ライフサイエンスに関心を持っていただければ幸いです。和賀さんから今お話がありましたようにですね、今後もですね、UC サンディエゴ校、そして慶應義塾大学のジョイントウェビナーをはじめですね、いろいろなウェビナーをいろいろなテーマで今後も開催していこうと思ってますので、何卒よろしくお願いします。今日はありがとうございました。
ワンモアメッセージいいですか？
岡野先生、手挙げていらっしゃったんですね。
最後日本語で、Please let me speak in Japan。この私のバック、ソークインスティュートって言いまして、本当に UC サンディエゴとすぐ隣にありまして、谷下玲也さんのビルはここから100メートルぐらい。only 100 meter from this view. Okay, so it's called the Sanford Regenerative Medicine Institute. 本当にここから100メートルのところに谷下玲也さんは、研究してます。で、こんなにも毎日太平洋を見ながらですね、研究してまして、もうこのソークインスティュートがもう、すっごい数のノーベル賞受賞者出てるんですよね、実はね。本当にここ来るだけでなんか非常にいい発想が生まれそうなところで、ジョナサン・ソークっていう人がリサーチをするためにどういうことを考えなきゃいけないかっていうこともちゃんとメッセージが刻まれてます。ぜひ言いたいことはですね、この慶応 UC サンディエゴウェビナー、ウェビナーだけじゃなくて本当に 近々、来年にはですね、インパーソンでやりたいと思いますし、そこにリンク J としても、インバルブしていきたいと思ってます。本当若い人たちにもそこで参加していただきたいと思ってます。私、実は慶応大学医学部教授ですが、リンク J の理事長っていうのもやっておりますんで、ぜひそっちの方もですね、やっていきたいと思ってます。今日は谷下玲也先生、さや先生、どうもありがとうございました。どうも、お付き合いいただきましてありがとうございます。では、
はい、ありがとうございました。このね、今、岡野先生からお話しいただいた総区因子っていう章、私も行ったことあります。素晴らしい研究環境ですし、素晴らしいビュー、夕日が特にね、美しいとこですね。皆さんね、ぜひね、こういったところで研究できるようにね、これからも、いろいろやっていただければなと思います。はい、誠にありがとうございました。じゃあ、ちょっと司会の、関さんの方に戻します。お願いします。
はい、皆様ありがとうございました。今後もリンク J と UC サンディエゴの共催ウェビナーを開催してまいります。最新情報はリンク J のホームページをご覧ください。また今後お聞きになりたいテーマや登壇者についてご意見ご要望いただきたく、ウェビナー終了後に表示されておりますアンケートより皆様のご意見ご感想をお聞かせください。皆様のお役に立つセミナーを開催したくご協力のほど何卒よろしくお願いいたします。また、リンク J のイベントお知らせメールを受け取りたい方は表示されております QR コードからご登録ください。その他のお問い合わせなどはリンク J ホームページよりご投稿いただけますと幸いです。皆様本日はご視聴いただきまして誠にありがとうございました。